The paper reports the results of a long-term pilot-scale trial where food waste as solid substrate was treated in a two-phase thermophilic anaerobic digestion process, optimized for hydrogen and methane production. First phase of optimization for hydrogen production was obtaining recirculating the effluent coming from the methanogenic phase and without the addition of external chemicals in order to exploit the residual buffer capacity of the methanogenic digestate. This is a less expensive alternative to the use of chemicals for external control of the pH in the phase of dark fermentation to maintain the pH within the optimal range 5-6 for the hydrogenase catalyzed reaction. A drawback of such approach is the recirculation of ammonia into the first phase reactor for hydrogen production, with possibility of consequent inhibition. Therefore, this study was focused on the development of a control protocol based on ammonia concentration. The first part of this paper illustrates how the use of variable recirculation flow makes possible to control the whole process, preventing the ammonia inhibition in the system. The main problem linked to this approach regards the choice of control parameters to be measured online. In fact, ammonia concentration probes in such heterogeneous media could be difficult to use and, on the long run, may prove to be not reliable. Thus, the approach used here considers to use an indirect measure of the main control parameter using simpler predictors. These indirect predictors can be also measured in easier way using online probes. As it can be seen, the two parameters, ammonia and electrical conductivity, are linearly correlated for both reactors. The measurement of the conductivity is therefore a good candidate in terms of control. In fact, it is a non-specific measure, measurable with strong and reliable probes which demand for relatively low maintenance procedures. Also, alkalinity showed a good correlation with conductivity, while VFA showed an inverse correlation. As final remarks, experiments done suggest how the control of inhibition can be based on ammonia content, and this parameter can be easily linked to the other, such as alkalinity, VFA, but especially conductivity, as shown by the chemometric analysis. According to the study, the experimental data of this evaluation are chemometric modeled for the reactor of methanogenesis, called F2. Important from the point of view of the possible prediction of the ammonia-nitrogen concentration in the recirculation flow. The equation of the multivariate model, using the predictor variables alkalinity and conductivity to predict the concentration of ammonia, is the following on slide title. In this regard, the graph about the progression of ammonia observed, the real value, was developed, in relationship with the one predicted by the latter equation of the multivariate model. The stability of the model is given by the values of square R and square Q, due to the external validation. The SDAP calculated represent good prediction of the model, as demonstrated in the graph above. This pilot-scale study shows that it is possible to obtain a stable hydrogen production by dark fermentation without physical or chemical pretreatments, when biowaste is used as solid substrate. The optimization was reached with only dynamic recycle of digested sludge from methanogenic reactor, which allows to maintain the pH at an optimal level for hydrogen evolution in the dark fermentation reactor. A stable bio-hythane production was obtained with specific gas production of 0.69 cubic meter of biogas per kilogram of waste fed. Comparing the predictive capabilities of the models and the economic feasibility, the best model seems to be the one based on two predictors, conductivity and alkalinity. This demonstrates that the use of electrical conductivity and alkalinity measured online could be the best model option for online monitoring of this process. As final remark, the development of a real semi-automatic control of the whole process using basic models able to predict the concentration of ammonia is developed and validated. This can be done using the online measurement of easily quantified parameters, allowing to the use of low-cost probes, reaching high-degree analytical strength at the same time.